brain is it's got the dura mater still on it. It's this opaque, kind of looks like plastic shrink wrap, and it really is tough. And a lot of the cranial nerves that come off of here are embedded within that dura mater. So if you just try to rip and tear that dura mater off, you're going to tear all the cranial nerves off as well. So what we've done in a couple of the bags is we've carefully cut away portions of that dura mater so that you can see the cranial nerves more clearly. So the dura mater, pretty easy to identify just because it's this opaque membrane, and it is kind of like shrink wrap. It doesn't fit really tightly, but it does provide a great deal of protection to the central nervous system. It's around the spinal cord as well as around the brain. So that's what the dura mater would look like. And also, just for curiosity's sake, this little round dewjobber right there, that's where the pituitary gland is. So that's what would sit in the sella turcica, that little pocket of the sphenoid bone. If you guys remember when you pop the top of the skull off and you can see that little pocket in there, there's the pituitary gland. But usually when you remove the dura mater, the pituitary gets taken off and it will be absent on almost all the other brains. So this is what they look like when we get them from the company that prepares these for us. When the dura mater is removed, it's kind of like this stuff. It's like shrink wrap, but it's really tough. The word dura is where we get the word durable from. So it's like trying to open up candy bags. You know, they're childproof. You know, I have to get scissors. I can't do it. But the dura, it really is tough material. All right, so there's just kind of some general brains to look at. Let's take a look at a whole brain. And there's only one in this bag right now because there were a couple intact ones earlier. And what I did is I was going to cut this in half. But I just want to give you a little bit of orientation of what the brain is like. So this brain came from a sheep, and if the sheep was standing on all four legs facing away from me, this upper region would all be the cerebrum. This little thing hanging off the back is the cerebellum. The spinal cord is the skinny little portion coming off posteriorly. If we flip it over, right where the spinal cord begins to expand outward and sideward, right at that spot, this is where what's called the brainstem begins. And the brainstem, the inferior-most portion, is the medulla oblongata. There's a pons, which looks like a little speed bump. All that dark stuff is just where a blood vessel broke and stained the tissue. So that's an artifact. Most of our brains don't have that spot there. And then some of the nerves are present on here, and they are these skinny little white things. We'll get a chance to look at some of them in just a short while. Actually, that's a pretty good brain. A couple things are missing on it, but we may come back and look at that later. Here's little remnants of the dura mater that are on the optic nerves where they come together. So this is what an entire brain would look like. And there are 3 layers of meninges. Have you guys talked about meninges yet with the spinal cord? There's what's called the dura mater, that stuff on the outside. And when that dura is off, all the stuff that has these blood vessels running around beneath it, the black, brownish-looking stuff are blood vessels. The next layer is called the arachnoid layer. And if that layer is removed, right on the surface of the brain itself, or the spinal cord, is what's called the pia mater, and that means the delicate layer, the delicate mother. The word mater means mother, so dura mater means tough mother. So mother is like nurturing, wrapping around it. Dura means tough, so it's a tough mother, and that's used in the vernacular as well as in reality. And then pia means delicate. Oh, it's closest, and a very thin little layer right on the brain itself. Right down the center of the cerebrum is called the longitudinal fissure, and I can actually put my finger in that little longitudinal fissure and kind of separate the arachnoid layer so I have 2 halves of the cerebrum. And it's true, the left half controls the right half of your body and vice versa, and then any sensation from the lower right half of your body, from below the neck, comes up, crosses over, and the left part of your brain feels that. So if you touch my arm, my left arm, my right side of my brain just felt that. Thank you. Okay. And if I kick you, it's the right side of my brain making me kick my left foot. And you better behave, or else I'll use the left side and kick the right one too. So, all right. So there's just some general features of the brain. I'm going to cut this one mid-sagittally just so you can see some of the internal structures. Then we'll get another brain out and look at some of the cranial nerves. Now, you won't need to do any sectioning, but when a brain is cut mid-sagittally, we get 2 halves. And I did such a good job of cutting this, I didn't get much of the septum pellucidum. Okay, here's a couple structures. You already saw the dura mater. Look at how the white matter just jumps at you. Doesn't the cerebellum look like a tree to you? So there's white matter, which is all wiring that connects the cerebellum to various other parts of the brain, and all the darker brownish grayer stuff is gray matter, and that's where all the muscle memory, where information is stored and acted upon. All this white stuff in here is also white matter, and that connects the left and the right half of the brain together. If I pull open this area, there's an opening right in there. That opening is the lateral ventricle. And you see that little remnant of membrane that's right there still? That's the septum pellucidum. And some of it is still in place right here. I mean, it's such a perfect cut, I cut right through it, so it's not on one half or the other. So the two of the three structures that we've added, we've already looked at. The dura mater, and now you've seen the septum pellucidum, the membrane. And if I take this probe and go fishing around inside that lateral ventricle, see that little dark brown thing that sticks out? Looks like a tongue sticking out between an upper and a lower lip. That's the choroid plexus. So if I would ask that on a lab exam, it's going to look like a little dark thing sticking out of the opening of the lateral ventricle. So the three structures that were not in your lab manual, you've now been able to see. The dura mater, the membrane, which is the septum pellucidum, and this little dark structure, which is the choroid plexus. We'll tuck him back in. Okay. All right, I'm not going to go through all the structures, but there's what a half brain would look like. So now that that one's been cut open, we're going to put him in the bag with the other half brain. And that's not meant as a slur, it's just meant the reality of that's what it is. So don't go home telling people I called one of these guys with half brain, because that would not be true. All right. This bag has brains that are whole brains that we'd be real careful to take the dura off, and we can see the cranial nerves that you need to identify with them. So let's pull one of these out and see if we can find most of the cranial nerves that we need. All right, the cranial nerve, just like the spinal nerves, are numbered. Can you peel off a piece of paper towel, Shelby, maybe about an inch or a foot and a half long, just so I have something to wipe my hands on and not flowing liquid all over you guys as I talk? Thanks. I was just looking at YouTube last night, and I think there's actually a YouTube video of me doing this. It's already posted.
Oh,
there.
that one's the heart.
There's one with the heart, there's one with the cap, there's one with the brain, too.
I didn't see the brain one.
Okay, yeah. Anyways, someone had said, I thought you were there. I looked it up last night, and I thought it was there, so maybe now these will get posted, too. Okay. So all the cranial nerves, if the sheep were standing upright, they are numbered from the most anterior to the most posterior and superior to inferior. So the numbers are going to start up near the front of the brain, and we're going to go backwards with them. So cranial nerve number 1. These little flaps here and here, those are olfactory nerves. Sense of smell right there. And these are huge. These are much larger than ours are in our brain. So smell is a much more important sense for sheep than it is for us. I mean, smell is important to us, but most other mammals it's much more important than it is for us. So there's the olfactory nerves. These big stumps that come in and form like a white cross or X, those are the optic nerves. And where they crisscross, right at the center, is called the optic chiasm or the optic chiasma. And chiasm or chiasma is Greek for cross. So optic nerves are number 2. Number 3, these little things that look like little flat pasta noodles, those are cranial nerve number 3, oculomotor. They help to move some of the muscles that move your eyeballs around. Oculomotor. Cranial nerve number 4 looks like a little piece of white fishing line, and it's situated between the cerebrum and the cerebellum. I'll pull back a little bit so that those of you can see it from the side. See that little white thing with like a little piece of white fishing line in there? Come up. That little thing right there. By the way, there's one on each side. That's called the trochlear nerve. Number 4. Cranial nerve number 5 is pretty good size. It is this large stump here, and it's called the trigeminal nerve. So it's located, this little bump is called the pons. So we've got the medulla oblongata, and then the pons. So the trigeminal nerve is located right on the sides of the pons. Number 5. By the way, these are bilateral. They're on both sides. They're paired. Same with the spinal nerves. Number 6 begins on the medulla and rests on the pons, and it's this much skinnier pasta. This is like angel hair pasta, and that's cranial nerve number 6, called the abducens nerve. When you abduct, an abducting motion, as you talked about with muscles, abducting means to move away. So if I'm looking at you with my right eyeball and I abduct my right eye, I just did an abducting motion. There's a muscle on the side of my eyeball attached to it. And when the nerve innervates that muscle and makes it contract, it attaches to the outside of the eye and it pulls it and makes my eyeball abduct that direction. Which means that if this eyeball is abducting, the other one over here has to relax, and one on the inside has to pull, otherwise my eyes will look different directions like a chameleon, and we don't do that. Ours are paired together. Anyways, that's number 6. Skinny little guy, abducens, begins on the medulla and rests on the pons. Number 7 and 8 are the last two. They rest right behind the trigeminal nerve. They're about the same width as this pin, and they are perpendicular. The trigeminal nerve comes off of the pons and kind of proceeds forward anteriorly. That's that great big stump right there. It's about the same diameter as the handle of this probe. Right behind it at a 90-degree angle are two nerves called the facial and the auditory. They're just two skinny little nerves side by side right behind the trigeminal. And now cranial nerves 9, 10, 11, and 12 are all this stuff down here that we're not going to get into because there's bits and pieces missing from them. Now 7 and 8 are so small, the way to remember them, and even on a lab exam, if I stick a pin right into one or the other, it's often difficult to tell which one I'm stuck in because they're so small. But if you can remember that your face is in front of your ear, facial is the more anterior, auditory is the more posterior. By the way, that nerve, the auditory nerve, has 3 names that are all acceptable. You can call it auditory, or acoustic, or vestibulocochlear, whichever one you want to use. The structures in your inner ear that are responsible for hearing and balance are called the vestibule and the cochlea. So vestibulocochlear tells you the names of the structures that those nerves originate from. You can call it acoustic or auditory because they both kind of come from the ear. So whatever term you want to use is okay. All right, so I've named them. You guys name them as we go through. Number 1 for sense of smell.
Olfactory.
Olfactory. Number 2 for vision. Optic. Number 3 to move your eyes. Oculomotor. Number 4, it's a little fishing line between the cerebrum and the cerebellum. Trochlear. By the way, this also moves one muscle around your eye. There's one muscle attached to the superior surface of your eye, goes up towards the nasal portion of the orbit, and it loops through a little piece of connective tissue like a pulley, and a trochlea is a pulley. So a trochlear nerve means it innervates one muscle that goes through a pulley to attach to the eyeball. So that's why it's called the trochlear nerve. Anyway, that's trochlear, number 4. 5, big stump, trigeminal. 6, makes your eyeball move sideways, abducens. And then 7 and 8. 7 is facial, 8 is auditory. So there's the 8 cranial nerves on the sheep brain you need to know. And because these are in really nice shape, we're going to keep these in that bag. So whatever bag you get a brain out of, just return it to the bag it came from, and that way it's still nice and fresh. Many of those other regions, I'm not going to go through all the different lobes and all the other sections you can see. Between the website you'll be able to utilize and your lab manual, you guys can find a lot of that stuff and then come back in on your own or use websites to help you as you do that. And then we'll, when we're done putting this stuff away, we'll talk about options for next week and then for your lab exam. When you're done looking at all the sheep brains can simply return to the bag which they came from. If you're working with these, you're welcome to get them out, put them in a tray, look at them, identify parts, but maybe after about 15 or 20 minutes out, they need to go back in the bag. And there is a little bit of liquid there, so that will help to kind of keep them hydrated. And then as we see need to, we'll take them out and put them in some of that liquid to rehydrate them. But don't leave a sheep brain out and then go run an errand for an hour and a half and come back, because when you come back, you'll have sheep brain jerky. It'll just dry up and get unusable. And these are the same brains we'll be using on your lab exam. So take good care of them and use them. They are fairly delicate, although once they're preserved, they're much firmer than real brain tissue would be. But you're using sharp probes with abrupt ones. If you're real abrupt with them and real, rough, you'll mess up all the tissues and the little nerves kind of scrambled. So just be careful when you